Again, I have to edit the title. I will edit later on. Don't worry.
Okay, sir. Shall I start then, sir?
Yeah, you can start. You can start.
Okay. Thank you, sir. Good morning, one and all. I'm very happy that in our PhD webinar series, today we are having our 10th consecutive speaker, Dr. Gita. And I welcome all the people who are watching the live stream, as well as the people participating in today morning. First of all, I would like to invite Dr. Arvind Deshmukh, All India President, Microbiologists Society of India, to speak few words, sir.
Thank you, Jay. Really, I'm so happy. The reason is that the young generation of India doing extensive research, but they are not getting name and fame. Why we should give name and fame to them? Not because they want, because we want. And it should be inspiration to all young scientists aspiring to do research, aspiring to do PhD. So it is our attempt. It is in short, a national service. And we are doing it. We are felicitating all the speakers with a certificate and memento from Microbiologists Society. Really, I appreciate Dr. Vivek Upasani, Dr. Prakash Halami, regularly attending such seminar and inspiring these young scientists. I welcome all and express my thanks to Jay also for organizing such a beautiful event. Thank you. Thank you.
Thank you very much, sir. Thank you. In the interest of time, I would like to introduce our today's speaker, Dr. Gitanjali Ganguly. Dr. Ganguly is currently serving as assistant professor at Sophia College, Mumbai. Prior to that, she has worked as assistant professor at Wilson College, Mumbai. Ma'am has done her PhD in KIIT Bhubaneswar. She has done her MSc in applied microbiology from the same university. Ma'am has done her BSc from University of Calcutta in 2011. Ma'am has received some very prestigious awards and grants to present her work. She has received Bill and Melinda Gates Travel Award for attending the 17th ICIT 2016, Hyderabad. Ma'am has received Guarant International Travel Award for oral presentation in San Diego, California in 2015. Ma'am has also received Foundation Marriott's Travel Award for oral presentation at IMRP 2015, Singapore. She has also been awarded with DST Inspire Fellowship for her doctoral studies, and ma'am has been awarded with the KIIT Merit Scholarship 2 times. Very good, ma'am. Ma'am has published more than 12 peer-reviewed research papers in national and international journals. She has done more than 10 oral and poster presentations. She has organized various conferences and workshops. So today I welcome our speaker, Dr. Gitanjali Ganguly. Over to you, ma'am.
Thank you so much, Deshmukh sir, and Jay, thank you so much for the introduction.
Thank you, ma'am.
I am extremely happy that Microbiologists Society of India has given me this opportunity. And I'm really thankful to all of the organizers, organizing secretary and the coordinator. So I'll just start sharing my screen. Please let me know if you can see my screen.
Sure.
I hope
it is visible, ma'am. Yeah.
Okay. Good morning, one and all present in the platform here today. So I'll start my presentation with the title of my thesis work that I've done. It is to study the role of Mycobacterium tuberculosis acetyltranserase in the regulation of oxidative stress responses in macrophages. So firstly, why is it so important that we address the issue regarding tuberculosis is because of the report that we see, which is reported by the World Health Organization. So it tells that almost 10 million people fell ill with TB and 1.5 million people die because of this disease. And the pathogenesis is really very interesting. It resides inside a immune cell, which is macrophages, which is usually designed to get rid of the bacteria, but it is a very intelligent bacteria and it survives inside the macrophages. So it has got some very key survival mechanisms. The first being the phagolysosome fusion inhibition. So as soon as the bacteria is inside the macrophages after the phagocytosis, there is a inhibition of the lysosome fusion. Thereby, it helps the bacteria to survive inside the macrophages. The second is the inhibition of acidification. How does it do that? It inhibits the proton ATPase pump, and so there is no acidification. The pH is not lowered, and so the bacteria is able to survive. There is a role of the tachoproteins after the phagocytosis process, which it also modulates. And of course, Mycobacterium tuberculosis is a pathogenic bacteria, so it has a lot of virulence protein, which causes its infection. In my study, in my PhD study, I have mainly concentrated on the oxidative radicals. So I have studied that how it protects itself from the host oxidative radicals. So before moving on to my presentation, I would really like to give you an overview like how the oxidative metabolism works after the infection is taking place. So once the bacteria is inside the phagosome after the phagocytosis, the host usually go in for doing a respiratory burst, and because of that we have a whole lot of cascade of oxidative oxygen radical forming. Now what the bacteria does is it tries to modulate its antioxidant system, as in enzymes like catalase or peroxidins, which ultimately block the autophagy, apoptosis, and cellular damage that we usually see after there is an oxidative stress. So it's a superoxide radical forming because of the presence of superoxide dismutase. We see the presence or the formation of hydrogen peroxide, and eventually it's water and oxygen. So what the bacteria is usually done, or what is reported so far, is that it modulates the formation of these antioxidants. Apart from my study, the earlier studies like protein like WIB-B, DOS-R, DOS-T are all modulating the oxidative pathway, but they have been modulating it in all different ways. Like the WIB-B proteins is mostly involved in the immune invasion and virulence, and the DOS-R mainly regulates the dormancy signaling, as in we know that once there is an active infection, with the help of Mycobacterium tuberculosis, it mainly enters a latent phase because of the dormancy. Now, next, a very important key player in the mycobacterial pathogenesis is its cell wall. And in the cell wall, we have the very, very known and maybe the notorious one is the mycolic acid layer, which is mainly responsible for all kinds of drug resistance, drug efflux, and then all modulation downstream signaling molecules. So it is a very key player. The entire MTB cell wall is a key player, but because of the mycolic acid, it gets an added advantage. So if we see how it interacts, there are a number of cell surface receptors with the help of which the Mycobacterium tuberculosis interacts with the host cell. So it is the TLRs, it is the mannose receptors, scavenge receptors, and so on and so forth. So in my study, I will be mostly talking about the mannose receptors. And so you can understand the protein that I would have worked on for my PhD study is a mannose-related protein. Broadly, if I want to divide my study, I have two objectives. One was the in silico characterization of a mycobacterial tuberculosis protein, which is RV3034. And then I went on for studying its effect on the oxidative stress inhibition. Now, why did I come to this one particular protein RV3034 is because we did a corne lectin catch. Corne is a naturally found lectin, and because of that we were able to find some mannose-related protein from the delipidated cell wall. So we delipidated the mycobacterium cell wall. Then we conjugated the cell wall with the corne. So whatever got selected was the mannose-related protein, and we did a mass spectroanalysis to confirm that what is the protein that we have got. So while comparing it with the available database of the nucleotide sequence, we found that it's a protein which is resembling to RV3034. That's how I came down to this one protein, which is RV3034. Now, we made two models. My entire study is mostly basic microbiology and immunology. So we had designed two set of models, one in the non-pathogenic model, which is mainly Mycobacterium smegmatis, and other was made in a pathogenic model, which is Mycobacterium tuberculosis. Now, for developing or constructing a Mycobacterium tuberculosis mutant, we basically used a vector system, which was pristinamycin inducible promoter system. Now, while we were doing this cloning into MTBH37RV, which is the pathogenic strain, we find that if we were completely deleting the entire gene from the sequence, we were not able to find any kind of transform colony. So we went back and checked our literature. We found that it is one of the essential proteins. That is, without the gene, or if you are trying to knock out the gene, there would be no colonies formed. So what we did is we went for designing conditional mutants. What exactly it is? We developed two mutants. One is the N terminus with the N terminus. Another is with the C terminus.
Excuse me, ma'am. I request everybody to please keep yourself muted.
Okay, thank you. So we had made two kinds of mutant, one with the N terminus and another with the C terminus. So what we did is we tried to induce the expression of RV3034 in presence of a pristinamycin, which is the selection marker for this. So as you can see that MTB RV3034 plus PPTR here is pristinamycin. So we found our band of interest, which is around the 900 base pair, which is this one, and where we found that we did not induce, as in we did not add pristinamycin in the media, the RV3034 expression was almost negligible. Then we developed our non-pathogenic model construct, which was in the Mycobacterium smegmatis, and we used the PSMT3 shuttle vector here. So this confirmed that there is an ectopic expression, as in we overexpressed the gene in Mycobacterium smegmatis, as smegmatis already has a similar copy of RV3034. So it was an overexpressed mutant. So initially, just starting with how the mutants work, and to make sure that because of the overexpression or because of the conditional mutant formation, we did not disturb the growth kinetics of the mycobacteria. So we did a growth kinetic study considering the wild type and the mutants that we had, both in case of H37RV and M. smegmatis. We found that up till 192 hours in case of MTV and 108 hours or 110 hours in case of M. smegmatis, there was no growth defects. So we confirmed that because of the expression or overexpression or any mutant formation, there is no defect which has occurred in the growth kinetics. Next, we went ahead to check for the mRNA expressions for the expression of the RV3034 in the in vitro condition. So after 0, 24, 48, and 72 hours, we isolated the mRNA from the cDNA and did an mRNA analysis. Same for Mycobacterium tuberculosis as well as for smegmatis. So we found that as and when the time is increasing, the expression for the media where there was pristinamycin added showed a higher expression, whereas where there was no pristinamycin added to the media, the RV3034 expression was almost same. Now, in case of smegmatis, also we see a time-dependent increase of the gene. So we concluded that in case of the H37RV, as adding with the pristinamycin, the expression of 3034 is induced. And in case of smegmatis, we do see a time-dependent increase. Okay. Then we went ahead for doing a tertiary structure prediction. This was all in silico prediction to understand that what exactly this protein was all about. It's a hypothetical protein, so we needed some lead to go on to channelize our study to a direction. So we found that it has got a conserved acetyltransferase domain. So in the first figure, we did a structural optimization. And in the second figure, in figure B, we did all kind of domain analysis study. So from here, we concluded that yes, it has a bacterial transferase hexapeptide repeat, which is a very, very prominent and a precise feature for acetyltransferase. Then we went ahead and checked for the acetyltransferase activity, and we wanted to check that how is it different from that of the wild type. So we took our wild type cultures in presence and absence of pristinamycin, and we took our conditional mutants. So you can see that when there is no pristinamycin added, then when there is no induction of the gene, so there was no acetyltransferase activity, which we took a colorimetric reading at absorbance of 412 nanometer. And the wild type and the RV3034 plus pristinamycin conditions showed a higher acetyltransferase activity. In case of smegmatis, we found that the overexpressed strain, which is MSM RV3034, MSM here stands for Mycobacterium smegmatis. So the overexpression showed a higher acetyltransferase activity with time. We also did went in for purifying the RV3034 protein, and then we found that the recombinant protein also showed a higher expression in comparison to the BSA, which was a negative control, and we in heat inactivated RV3034 protein, and then went ahead for checking the enzyme activity. Now, this was a very preliminary experiment that we have done to just see that what could be a probable substrate for the acetyltransferase, because when we went ahead and checked for the literature, we found that such similar proteins were mainly sugar acetyltransferase. So we took a range of sugars, and then we found that maltose showed the highest substrate specificity. These were all about its amino acid compositions and secondary and tertiary prediction. So we found that from here, our protein consisted more of the hydrophobic amino acid, and there were more of loop regions. So it is mainly involved in cell surface, increased cell surface hydrophobicity, which was a very interesting result that we got, because I mean, if a protein or a pathogenic protein which has got more hydrophobic amino acid, which has got more cell surface hydrophobicity, would definitely be a very tough protein in case of its pathogenesis, as in the bacteria would really like to modulate or channelize those protein towards the pathogenesis. Okay, and just as since we are talking about Mycobacterium tuberculosis, drug resistance is always there in the back of our minds. So we just wanted to check that did it had any role in checking for the drug sensitivity. So we took some first line drugs like isoniazid, pyrazinamide, and ethambutol, and all showed resistance, as in the bacteria was not susceptible to these drugs. So this was another interesting fact, which the lab is now carrying on the work on it. Okay, now since we are talking about pathogenesis, finally we came up that since it holds so much importance, like it is hydrophobic. I mean, imparts more cell hydrophobicity. It has got, it showed resistance towards the drugs. So what about the survival? Is it enhancing the intracellular survival? So what we did, we took some mouse macrophages, which was raw 264.7 cell line, and then we infected it with the mutants that we had. One is the overexpressing strain of in the smegmatis, another the mutant, and we see that both in case of MTB and MSM, the survival increased in comparison with the wild type or the vector control. And if you look into the minus PPTR condition, where there is less 3034 expression, the intramacrophagic or the intracellular survival of RV3034 strain came down. This suggested that yes, it showed significantly higher intracellular survival in comparison to its wild type. Very interesting. Now, how is it surviving? What is it modulating? In my beginning of my presentation, I told that I was mostly interested in oxidative stress. So we move ahead with checking the ROS. What's the status of the reactive oxygen species post-infection. So we did DCFHDA staining, and then we went ahead with the flow cytometry. So we found that in comparison to the control cells and to the vector control, the control are mostly the unstained cells where there is no infection. So the healthy cells are mostly on the left side of the panel. And when there is the infection with Mycobacterium smegmatis, PSMT3, which is my vector control, we see that there is a huge shift towards the FITC positive axis, which claims that yes, there is oxidative stress. Now, if you see that when there is a infection with the overexpressed strain, that is RB3034 in smegmatis, there is reduction in the FITC positive cells. That is, it indicates that there is less of the ROS formation. So we can, it gives this entire experiment gives an indication that yes, with the overexpression of RV3034, we see a inhibited ROS formation. Now, we went ahead with inducing ROS with a chemical known as Zarmosan. It's a ROS inducer, and then we saw that what when the infection is there, and we are also inducing oxidative stress from outside. Now we see that there is a dose dependent increase, but when we compare it with our vector control, it's mostly inhibited. So even after the ROS is induced in the cells with the help of zymosyne. RV3034 is so potent and so good at its work for inhibiting ROS in the microenvironment that even after the induction, there is a very significant decrease in the ROS formation. Obviously, at a very high concentration of 250 microgram per ml, it did not show that inhibition pattern because of the high load of zymosan. So we concluded that RV3034 showed a dose-dependent accumulation. So as in when the increase is there, though it's inhibiting, but at the final concentration, it's not able to retain that. Now, the next phase of my study comes where we actually wanted to see that what actually is helping, or what is the mechanistic insight, that how is this oxidative stress is getting inhibited. So here, I would like to draw all your attention towards a term which is called as peroxisomes. Usually, when we talk about oxidative stress, the first thing that comes in our mind is mitochondria. But here we have tried to explore another organelle, which is kind of similar to mitochondria, that is peroxisomes. It is mainly a organelle in the host cell, which allows the beta fatty acid oxidation of the very long chain acids. And this actually, the thought of acetyltransferase, oxidative stress, acetyltransferase is a class of enzyme which mainly helps in the beta fatty acid oxidation and all, where we came up with a thought process that yes, let us just explore a similar kind of molecule, and we landed up studying about peroxisomes. So just a brief introduction what peroxisomes are. It is a small membrane closed organelle, and it is known as the oxidative organelle. Like I said, it helps in the long chain fatty acid oxidation and lipid peroxidation. So it is very, very vital for the cellular process. The only study that we could trace back was a study which was done in chlamydia. And we found that that study says that these chlamydia species takes up or hijacks the peroxisome and helps, or it modulates the peroxisome in such a way that it helps in its intracellular persistence. So we got a very good lead over there, though very few reports were there, but we got a very good lead. So if we look on to the biogenesis of how is it formed, so it is mainly coming out or budding out of the endoplasmic reticulum, and there is a number of peroxin proteins which we denote as PECs. So there are a number of peroxin proteins which are followed all through the pathway, which helps in this biogenesis. So first there is a pre-peroxisome formation, then there is a mature peroxisome formation, and finally we have the division and proliferation. Okay. So we mostly did Western blot analysis to confer whether there is a biogenesis happening, and what if there is a less expression, what is happening to the peroxisome biogenesis. So the first set that you see over here, PEX3, PEX5, PEX11, is all related to peroxisome biogenesis. These peroxin proteins are all associated with the formation of peroxisome. So you can see that where there is expression of the RV3034, we have overexpression of these peroxin protein. These are the enzymes that are relevant to peroxisomes. So here also we can see that in case of peroxisome RV3034 expression, the expression of acyl-CoA oxidase and other enzymes are also increasing. We also studied about a nuclear transcription factor, which is very important in case of peroxisome. It is a peroxisome associated nuclear protein, PPR gamma, and the expression was higher in this case also. Now, I already introduced to the term zymosan. We continued our experiments with this ROS inducer, and you can see that the expression is coming down when there is induction of induction of ROS formation. So which led us to the conclusion that this RV3034 overexpression in the mycobacterial species is helping in the peroxisome biogenesis and proliferation. So these are biogenesis, and the entire pathway that we are going to see forward would be more of proliferation. Now, the previous results were mostly in the MTB model, that is the pathogenic model. This is the non-pathogenic model. We have checked for similar sets of gene, and which we found that with the disruption of peroxisome formation, I'll introduce to a new term here, which is TZ. TZ is a chemical which helps in the peroxisome inhibition, as in it stops the peroxisome biogenesis. Now, when there is no peroxisome inhibitor, I call TZ as a peroxisome inhibitor. So in absence of the inhibitor, there is overexpression of all the genes. In case of overexpress RV3034, and as and when there is inhibition of the peroxisome biogenesis, the function is not retained. So from here, we further get a confirmation that yes, RV3034 is involved with peroxisome biogenesis. We did an RT-PCR analysis for two very important enzyme related to peroxisome division and the formation of the new peroxisome, which is FIS and DLP. One, we did it in presence of zymosan, and the other we did it in the presence of the TZ, which is the peroxisome inhibitor. And finally, we could conclude that, yes, you can see that when there is overexpression, there is higher of the FIS and DLP, the proteins that are telling that, yes, the overexpression of RV3034 is helping from the mature peroxisome formation of the daughter peroxisome. Now, when there is inhibition, there is an inhibition or a decreased mRNA fold chain. So we say that disruption of the peroxisome formation resulted in its decreased biogenesis in the Mycobacterium RV3034 infected cells. So these are similar results where we studied about the biogenesis in using the immunofluorescence antibody staining. So if you can see that there is more of this puncta staining here. So usually when we see biogenesis, there is more of this puncta, that is the more of these red dots. Whereas when there is the addition of the TZ, that is the peroxisome inhibitor, we see that the punctas are reduced, or there is no active red specific dots here. This gives us a further confirmation. Now, since we are talking about its biogenesis, it is very important for us to read about how is it modulating the proteins which are mainly related to pexophagy. Here, another word, let me introduce. Pexophagy is the autophagic degradation of the peroxisomes. Since it's oxidative stress, so we have to read about autophagy. And since the model organelle that we are using here is peroxisome, so we would be telling it as pexophagy. So PEX14 is one of the first initial pexophagy genes, pexophagy related genes. And we found that in case of the overexpression of RV3034, there was a very distinct inhibition. And when we were inducing ROS in the condition, or when we were inhibiting the peroxisome, there was an increased expression of PEX14. So which we further concluded that RV3034 downregulates the expression of PEX14, that is, in its usual overexpression or infection, it is inhibiting the pexophagy process by inhibiting the very first molecule, which was PEX14. Now, downstream to that, we read about two factors, very important factors and transcription factors of the pexophagy pathway, which was P62 and NBR1. So in case of the RV3034 express strain, it was less. So since there is less of P62 and NBR1, there is less, we could expect that there is less autophagy happening. But when there is induction of ROS using zymosan, the expression is very high, significantly higher than that of the untreated one. So this was the same thing with the MSM model. So from here, we concluded that it is down-regulating the pexophagy pathway via subsequent interaction of P62, NBR1, and PEX14. And the very famous autophagy protein is LC3, 1 and 2. So if we all know that if there is a LC32 formation, there would be higher autophagy, higher is the autophagy happening inside the cell. So we found that there is more of the LC32 ratio in case of the less expressed RV3034, and the balance between LC31 and 2 is the same when there is more induction of RV3034, the same in case of the smegmatis model. So which claims that yes, if there is a down regulation of PEX14, there is subsequent less interaction of P62 and NBR1, and so the organally down regulating the autophagy, because there is no LC32 formation. No LC32 formation, there would be definitely an inhibition. And the same reverse phenotype is observed when we are inducing the condition with zymosan, or we are treating the cells with the peroxisome inhibitor. Now, slowly further, we moved upstream of what we are going to decide. So we went up to check for another autophagy related protein, which is like upstream, which is phospho-AMPK alpha. To our hypothesis, to support our hypothesis, it also showed that yes, it is less in case of the MTB RV3034. Overexpressed, and higher when there is less expression of RV3034. We then further studied about the mTOR. mTOR is also a very relevant protein when we are studying about autophagy and oxidative stress. But it is like that if we have more of mTOR phosphorylation, then the autophagy is down-regulated. So we checked for the phosphorylation of mTOR by doing Western blot analysis. And we found that with the overexpression of RV3034, the mTOR is showing higher phosphorylation in comparison to that of the inhibited or the mutant strain. Same was seen in case of peroxisome inhibitor and in the MSME model. So you can see that there is overexpression of phosphorylated mTOR, and here we found an less expression. So all these corroborated to our studies that yes, RV3034 is blocking the pexophagy pathway, and it is reducing the oxidative stress, and thereby it is helping the mycobacteria to grow. We have studied a number of other autophagy related proteins like ATG7, ATG5. We tried to study the transcriptional factors like TFE, BKEP1, NRF2, and from all this Western blot analysis, we found that RV3034 modulated the pexophagy pathway via AMPK, mTOR, and other autophagy related genes. Now,
excuse me, ma'am.
Yes.
I think there is some connection issue.
Are you not able to hear me?
Yes, now it is clear. Sorry, please continue.
Okay, no problem.
So not properly audible, ma'am. Actually,
yeah. Am I audible now?
Yeah. Now it's
okay.
Yeah.
Okay. Okay, sir. Okay, thank you. Now here, since we got a hint that mTOR phosphorylation was helping RV3034 for its autophagy in reduction. So what we do in the next subsequent experiment, we use mTOR inhibitor, which is rapamycin. Rapamycin is an mTOR expression inhibitor. And then we checked for the autophagy related genes after RV3034 infection. So we found that once there is uh, inhibition with the rapamycin, the expression was significantly less. So there was less phosphorylation. As a result, there was more autophagy. Now, how did we concluded that there was more autophagy? We check for the related proteins. Again, we check for ATG5, ATG7, and ULK1. Now, okay, we can understand that it's like RV3034 infecting the cells. It is modulating the ROS. The ROS is being modulated mainly inside the peroxisomes, and the peroxisomes are maintaining itself by not getting into autophagy. Who is inhibiting that? The main molecule that is taking part is mTOR. Now, so as soon as we are inhibiting the mTOR, the organelle is getting channelized into going into the pexophagy pathway. So we further confirmed it with the LC3, and obviously, since we are talking about pexophagy, we have to go in for checking PEX14. So we found that with the rapamycin, addition of the rapamycin, there were more expression of PEX14 here. We also check for PPAR gamma. So we see that this very important in nuclear factor for peroxisome is also getting down regulated when there is less of mTOR phosphorylation. Also the biogenesis protein PEX14, if you see these two expressions, there is significantly down, it is significantly down regulated. So we concluded that MTBRV3034 is regulated, the pexophagy process is regulated in a mTOR dependent manner. Now, since we are talking about mTOR and since we are talking about peroxisome, so we wanted to check that what if the nuclear factor, which is the PPR gamma, what if this is silent? Because there should be some link between mTOR and the pexophagy coming together in the peroxisome. And that linking factor, what we came across by reading the literature, was the PPR gamma. So we went in for doing siRNA of PPR gamma in the mouse macrophages itself. And then we infected those silent cells with the RV3034. And we found that, yes, that is where if you are modulating the expression of PPR gamma, that is with the less expression of PPR gamma, there is less phosphorylation of mTOR and there is more pexophagy formation. So here you see with the LC3 and the PEX14, we see an increased LC32 and of course an increased PEX14 in comparison to that of the untreated cell. Now the biogenesis peroxin, which is PEX3, you can see that when there is a silencing of PPR gamma and post-infection, we see there is a down regulation, whereas it is overexpressed when there is PPR gamma expression. So, and then we checked for the other autophagy related gene to see, to actually confirm that if we are blocking PPR gamma, which is at the topmost, and what is the downstream thing. So we confirmed that yes, all the autophagy related transcription factors and autophagy related genes were all getting down regulated. So we finally got a link that what happens after the post infection and how is it actually going and correlating. So we finally able to conclude that RV3034 regulated the peroxisome homeostasis. I want to stress into homeostasis because we are looking into the oxidative stress, and we know that when there is an oxidative stress condition, there is an imbalance in the homeostasis. So if we are not disturbing the homeostasis, how is it not disturbed? It is not disturbed via the mTOR PPR gamma dependent pathway. So if I want to discuss or summarize my study, so what is usually happening is we have the MTBRV3034. which is the protein of interest, it is getting infected, it is getting phagocytosed, then it is increasing the peroxisome biogenesis. As and when there is increase, there is expression of all the peroxisome related genes, that is PEX3, PEX19, 11. We found that there is higher expression of the proliferation and the segmentation genes also, like FIS1, DLP1. But when there is an induced oxidative stress, as in we are uh, exogenously inducing the oxidative stress, then everything goes in veil, like there is more of exophagy formation. Now you would say that why is it then inducing a exophagy over there? If we have uh, more of any organelle which is not in use, then it is a metabolic burden for the cell. And but the bacteria has a sort of mind that if we target something, like if we want to make a homeostasis, we have to kind of utilize the host's metabolic power, right? So it kind of quickly induces the pexophagy, so that there is loss of the disturbed or the non-functional peroxisome, and there is more of a homogeneity or a more of a metabolic homeostasis that is being established. So at the end of whatever MTB RV3034 is doing, is mainly targeting itself towards the thought process that we have to survive inside the macrophages. So yeah, firstly, it is helping in the inhibition of oxidative stress. But even if there is an oxidative stress, what is it doing? It is inducing the pexophagy a little bit higher than that of the wild type, because if there is more autophagy, there would be more clearance of the non-functional organelles. As a result of that, there would be more metabolic machinery or the metabolic enzyme that would be useful for the bacteria to take up. That's how we feel that the MTV RV3034 overexpressed bacteria are working. So this is the summary of my work. So we have characterized the manocylated protein after identifying it with the corneal lectin. Then it is one of the essential genes of the mycobacterium genome. As for those reasons, we had developed or constructed a conditional mutant. Then we check for its intracellular persistence. How does it survive inside the cell is with the inhibition of the ROS accumulation. What is helping in the ROS accumulation? It is because of the increased peroxisome biogenesis. If there is an increased peroxisome biogenesis, it inhibits the induction of exophagy, that is self-degradation of the peroxisome. And how is it doing that? It is doing that by modulating the mTOR PPR gamma pathway. So as per the future study is concerned, we can study about how mycobacteria and peroxisome are related during this infection process. The study was mostly done in the secondary cell line. So maybe we can validate the same using some real-time TB patient sample. And also, since we have oxidative stress in mind, so the granuloma formation could also be studied with respect to peroxisome biogenesis. Thank you so much. I am deeply obliged to the Microbiology Society of India, to Deshmukh sir, Jai sir. Thank you all for giving me this opportunity to present my PhD work in the Gujarat chapter. Any questions, I'm very happy to attend.
Thank you very much, ma'am. It was
Thank
an
you.
excellent presentation.
Thank you so much.
I think we already have a few questions in the chat box.
Okay. Do I need to go back and look, or maybe somebody
No,
can help
I can read out for
Okay,
you.
okay, sure.
Dr. Halami is asking how new targets can be identified to inhibit MBT based on this study, RV3334C.
How many? Sorry.
Yeah, I think Dr. Halami can ask himself.
Yeah, it was actually a wonderful presentation by Dr. Gitanjali. What I wanted to identify was now if you are to make any treatment against a mycobacterium, can acetyltransferase be used as a one target?
Yeah. See, we have number of acetyltransferases,
Yes.
like we have choline, we have sugar, we have histone acetyltransferase. Now, mostly this acetyltransferase is not that well studied. Why is it a potential target? Maybe we can design some drug targets, or maybe we can repurpose the already known targets. Is
Exactly.
because this will go and uh, affect the metabolic pathways. Usually these histone acetyltransferase or the other transferase are mostly related to the epigenetic ones, so they are not permanent ones. Maybe over the time period again resistant might develop. But if you are targeting any enzyme which is actually related to the metabolic process, then it's a more potent one. So yes, we do believe it's a potential drug target. We
Okay. So
in my juniors have also got some very good results
the,
further when they have done studies with the mice model. Yes.
yeah, the another just one more
Yep.
some general question was now any drug which you are actually using against mycobacterium, you think that they have to really penetrate inside the cell or without penetration also they can work? What is your opinion?
Penetration.
Yeah, because you showed that the cell
Yeah.
wall of mycobacterium is quite thick, rigid,
Yes.
and
Yes.
has several layers.
Yes.
So actually, when we were talking about our protein, which actually inhibits the RNA biosynthesis, and one of the structural biologists says that when the protein molecule is bigger, how you expect that it enters inside the cell. So that is what actually I just wanted to
Yes.
know
Yes.
your
So
opinion.
yes. So actually, synthesizing drugs, or even when we have synthesized lipopeptides or
Okay.
glycopeptides against the disease, what we have found was that the size was a very important factor. Now, as a alternative, what we had done a liposome. We have added a liposome adjuvant,
Okay.
which more likely was what it does was it was more likely to go and sit on the mycobacterial cell, and the uptake was easy rather than getting out of the cell or the cell pushing out the drug outside. So we have to design, or we have to employ such conjugates which are more like alike to that of the mycobacterium cell wall, because until and unless we have a drug which can penetrate inside the macrophage, is very difficult for the bacteria to get killed.
And there is a correlation between the peroxisome biogenesis and mitochondria, you mean?
Yes, definitely. There is not only a correlation of its biogenesis, its function is also kind of correlated, both works in harmony. Like in mycobacteria, it's mostly short chain fatty acids which are metabolized. In peroxisome, it's like short chain gets converted, long chain gets converted to short chain, and then that acetyl CoA short chain is further moved back to the mitochondria. So that's how it's interrelated.
And then PPIR gamma, which you showed, actually PPIR gamma is associated with obesity marker also. What is the linkage, by the way, here?
So PPR gamma has got it has got a very broad usage. Like we see PPR gamma in infection, we see PPR gamma in diabetes, obesity, cancer.
Exactly.
So PPR gamma is one of the very crucial nuclear transcription factor for peroxisome biogenesis. So if we look into the functioning, like how the biogenesis actually starts from the endoplasmic reticulum, it is at that very point we get the PPR gamma expression. That's why we intend to check that how is it functioning when we are doing an RV3034 overexpression. What is its effect on that?
Wonderful. Very nice studies. Actually, most of the studies which you are telling are the notable findings, I would say. but somewhere we were unable to see any, means the list of publications. Definitely you must be having a much more publication, but it would have been actually much useful. And I think PhD students should take this one as a example, as a benchmark, I would say, as Dr. Arvind sir already has informed in the group that we were expecting more students to attend this, especially the PhD scholar. But we see a little bit less attendance. I think there is another lecture is coming up soon from Microbiology Society. So congratulations once again, Dr. Gitanjali, and good luck to you.
Thank you.
From my side. Yeah, thank you.
Ma'am,
Yes.
ma'am, I'm Sriram. I'm doing my master's in microbiology, ma'am. So I have a query, ma'am.
Yes.
So I'm very much interested to study about mycobacterium. So, but in our university, we don't have a class 2 biosafety cabinet. So it is very tough to work with my pathogenic. So you have said that Mycobacterium smegmatis is non-pathogenic. So it will be safe to use for our study about the cell wall.
For the H37 RV experiments, I too, in my institute, I did not have that opportunity to work on that. So we also worked in a lab of a collaborator. But the smegmatis model, Mycobacterium smegmatis model, it has got a very good homology between the actual strain and the non-pathogenic strain. So you can work with it, but you need a BSL to laminar flow for that. I mean, you cannot really culture it outside like we do it in case, like normally we do it like E. coli or staph. We can do it like outside in the lamina, like between the flames or so. But because smegmatis nowadays have been reported to be opportunistic, so I would suggest that BL2 LAF is required.
Okay, ma'am. Thank you, ma'am.
Thank you. Somebody raised hand. I'm sorry.
Yeah, Preeti, would you like to ask a question?
Ma'am, is it plasmid bond?
I'm really sorry. I was to get that question. Last.
Nilima ma'am,
Are we
I'm sorry you were not audible.
I'm sorry, I'm not able to hear her.
Nilima ma'am, I'm so sorry. I think that
Hello?
connection issue.
If it's okay, she can type the
Yeah,
type
if
her
you
question. I
Okay,
can.
okay, no problem. No problem.
While do we have any more questions? Please feel free to ask. Because time is very limited. We have another lecture coming up very soon.
हो पाणी कमी झालं ना वरचं पाणीच नाही वरून लक्षच देत नाही.
Ma'am,
Yes.
ma'am, is there any other mycobacterial species that we can be working without the biosafety cabinet?
I am really not very in favor of working with any of the strains like outside the lamina, because there are no reports that any mycobacterium is very safe to use outside the lamina, like even when there are two burners on and you cannot just do a culturing of that. So I'll really not favor working with mycobacterium outside the laminar.
Correct.
Yes, ma'am. Okay, thank you, ma'am.
Thank you.
Thank you, ma'am. Thank you, Jai sir. Deshmukh sir. Thank you.
Thank you, Nilima ma'am. I think you are audible now. If you can ask a question now.
No, I wanted to ask, ma'am, is it plasmid bond RV3034?
Plasmid bond, as in, I'm sorry, I'm not able to understand what exactly you're asking.
Is the genome is plasmid borne? I wanted to ask.
No, it's a cell bound or a cell associated protein of mycobacteria.
Okay, ma'am. Okay. Okay, and ma'am, nice. You have hypothesized so many things like maltose for higher specificity, acetyltranserase, and PEX protein, how it has been down-regulated, peroxisomyogenesis. We really like, ma'am. And for ROS induction, significant decrease in ROS, as we noted. Thank you, ma'am. Great work.
Thank you so much. Thank you so much.
Yeah.
Thank you, ma'am. Dr. Just please type your email in chat box. So if we have any further questions from the audience, they can just take it up with you.
Yeah. You want me to put it on my chat box?
Yeah. Okay, perfect, ma'am. Thank you.
Thank you so much. Thank you so much.
Thank you, everybody. Thank you, Deshmukh sir, for providing us this opportunity and this platform. Thank you, ma'am. It was very much awesome to hear from you.
Thank you so much for giving the opportunity. Thank you so much.
Thank you. Thank you. Thank you.
Thank you, everybody. See you on next lecture. We have another one at 11:30. Thank you, sir.